Hello everyone, we're here to talk about electrophoresis, the definition, types, which are polyacrylamide gel electrophoresis and agarose gel electrophoresis, how to prepare the sample and how to run polyacrylamide gel electrophoresis and agarose gel electrophoresis, and the steps taken to run electrophoresis. Watch and learn.
Hi everyone, have you ever wondered how scientists identify animals from a single strand of hair or diagnose genetic disorders before symptoms appear? The answer lies in a powerful technique called electrophoresis. So today I'm going to be explaining electrophoresis, the types of electrophoresis, and how it works. So what exactly is electrophoresis? Electrophoresis is a laboratory technique that separates molecules based on their size and electrical charges. Think of it like a molecular racetrack. We use electric currents to make charged molecules move through the gel or other medium. The key factors here are DNA, RNA or proteins, all of which contains charged molecules. It is a very important technique in biochemistry. We use it for DNA fingering, in forensics, analyzing proteins, diagnosing genetic disorders, and even developing new medicine. There are several types of electrophoresis, each for its specific uses and applications. First, we have gel electrophoresis. It uses gel matrix as its separation medium. It has two varieties: agarose gel electrophoresis and polyacrylamide gel electrophoresis. Secondly, we have the SDS page, which means sodium dodecyl sulfate polyacrylamide gel electrophoresis. It is specifically designed for proteins. Thirdly, we have the capillaries electrophoresis. It is a more modern, advanced technique. Instead of using gel slab, it makes use of thin glass tubes called capillaries that has buffer solutions in it. Finally, we have paper electrophoresis. This is one of the older techniques where we soak filter paper into a buffer solution, which is used as a support medium. So in recap, electrophoresis is a laboratory technique that separates molecules based on sizes and their electrical charges.
Agarose gel electrophoresis is a technique used to separate DNA fragments and other macromolecules such as RNA and protein based on their size and charge. Now, the first step for DNA fragment separation is gel preparation and gel casting. An appropriate amount of agarose powder is weighed out. For a standard agarose gel electrophoresis, large amounts of molecules are better separated using low concentration, while smaller molecules are separated using high concentration. Now, note that agarose does not dissolve. You need to heat it. Then, while you heat it in a microwave and allow it to cool for about 55 degrees centigrade in a water bath, while it is being cooled, while it is cooling, a gel electrophoresis tank is being set up that consists of positive electrode and negative electrode. Then positive electrode and negative electrode. A column is being placed inside the gel electrophoresis tank. The reason for why the column is being placed inside a gel electrophoresis tank is for loading a sample. Then a casting dam is also being placed so that the agarose powder does not flow out from the gel electrophoresis tank. Ethylene bromide is also added. Ethylene bromide is highly toxic and should be handled properly. Now, ethylene bromide is interconnecting reagent. It separates DNA-based pair from bonds. Then, when it separates DNA-based pair from bonds, you pass it through a UV light. The more DNA pair is present, the more brighter it becomes. Then you allow the agarose gel solution to be poured into electrophoresis tank, and the gel is allowed to solidify at room temperature. Once the gel is solidified, the combo is carefully removed from the well and as a casting dam. The next is the gel is submerged in a TBA or TEA running buffer, which is used to provide the pH as well as the buffer.
Second step of preparation of agarose gel electrophoresis is
preparation of the DNA sample. Here, the loading buffer is being applied to the sample. This loading buffer contains bromoethylene blue dye and glycerol. It is being added to the sample and it is being allowed to sink down to the gel. This makes it easy for loading of the sample and for easy separation. After this, a molecular weight size marker or a DNA ladder is being added to the well. Well is being formed when the comb that was being placed on the gel is being removed. Well make it possible for easy loading of sample during the loading process. Now after the molecular weight size marker has been added to the well, we now proceed to add the samples containing bromoethylene blue dye and glycerol to other wells. Now add other samples containing bromoethylene blue dye and glycerol to other wells. Then a lid is being covered on the electrophoresis tank. Electric current is being applied to pull the samples across the gel at different speeds, sizes and charges. Now, smaller molecule fragments move faster than large molecular fragments. After the complete migration of these samples from the gel, the DNA fragments will be placed on a UV light and it will appear as a distinct color band. We'll now be able to compare our DNA ladder and the DNA sample band and our DNA ladder for easy approximation of the sizes.
Polyacrylamide gel can be seen as a liquid used to separate protein according to the size, in which polyacrylamide acts as metric, which a protein can be migrating through electrophoresis is used to separate protein as migrated through the polyacrylamide metric by the application of electrical current. Therefore, polyacrylamide gel electrophoresis has many steps.
Step 1 of polyacrylamide gel electrophoresis is preparation of the apparatus and loading of the gel. Here, you assemble the gel apparatus with gel plate. Place the gel assembly into the electrophoresis tank. Pour in the running buffer into the upper and lower chamber. Ensure that the entire gel is submerged to complete the electric circuit. Carefully remove the comb. When the comb is being removed, you can now raise the well that was being formed with a syringe containing running buffer. Thank you. Step 1 of polyacrylamide gel electrophoresis is preparation of the sample and loading of the gel. Assemble the gel apparatus with the gel plate. Place the gel assembly into the electrophoresis tank. Pour in the running buffer in the upper and the lower chamber and allow it to submerge with the gel to complete the electric circuit. Carefully remove the comb. If the comb is being removed, it will form wells. Then these wells make it possible to hold samples during the loading process or the separation process. This will make it possible to hold sample during the separation process or the loading of the sample process. Then use syringe to raise the wells with running buffer. Thank you.
Step 2 of polyacrylamide gel electrophoresis. Load samples. Prepare your protein samples by mixing them with a loading buffer and heating them to denation the proteins. Pour the samples into the wells using a micro pipette, taking care not to puncture the bottom of the wells. Then place a molecular weight marker known as the ladder in one of the wells for size comparison.
I'm here to talk on the step 3 of the polyacrylamide electrophoresis. While running the gel, place the lid on the electrophoresis tank, ensuring the electrodes are correctly aligned together. Connect the red and the black wire from the lid to the corresponding terminals on the power supply. Set the power supply to a constant voltage and start the run. Run the gel until the dye font, usually bromophenol blue, has traveled to a desired position near the bottom of the gel. Thank you.
Okay, I will be talking on the fourth step on how to prepare the polyacrylamide gel electrophoresis. So on the first step, we should disassemble and analyze. Once the run is complete, turn off the power supply and unplug all the wires. Carefully remove the lid and lift the gel apparatus out of the electrophoresis tank using a spatula or a gel releaser. Then you gently separate the glass plate to remove the gel without stretching or tearing it. Then you proceed to stain the gel using a commerce glue. And then proceed to distain the gel. The protein band becomes visibly clear. If using a pre-stained or stain-free gel, you may proceed to image it directly without an additional staining. Then finally, Analyze your results by comparing your sample band with that of the molecular weight meter to determine the size of the protein.
Hello everyone, hope you learned something about electrophoresis, different types of electrophoresis such as polyacrylamide electrophoresis, agarose electrophoresis, and the different steps used in preparing them. Thank you for watching. See you next time.